So I will talk about the gene expression, also known as the central dogma, uh, briefly. So we know that DNA is the genetic material of all organisms on Earth. When DNA is transmitted um, from parents to children, it can determine some of the children's characteristics, such as their eye color or hair color. But how does the sequence of a DNA molecule actually affect a human or other organism's future? For example, how did the sequence of nucleotides A's, T's, C's, and G's, adenine, thymine, cytosine, and guanine, um, in the DNA of Mendel's pea plants determine the color of their flowers. So I mean, we're asking this question that how we know that DNA transforms this information, but inside the body, how this information becomes that property. So we're talking about this, and this is called gene expression, and it is simply reading out reading out the information on DNA and producing the so-called properties like eye color, hair color, or the structure of the liver, the structure of some internal organs, etc. All kinds of information that you can ask yourself. A DNA molecule, isn't just a long, boring string of nucleotides. Um, Instead, it is divided up into functional units called genes. Okay? Each gene provides instructions for a functional product, that is, a molecule needed to perform a job in the cell or a type of protein, etc. In many cases, the functional product of a gene is a protein. For example, Mendel's flower colored gene provides instructions for a protein that helps make colored molecules like pigments in flowers petals. Okay, so there is this information in the DNA. Okay, it's called gene, and but how this gene appears in the body is that there is a specific kind of product which is called pigment protein, and just read out by some other molecules inside the cell, and as a result of reading, there are also machines that produces whatever the product that needs to be produced. In this example, it's called the pigments, and these pigments are produced and gives the color of the flower. If it's purple, if its parents is purple, then it's going to be purple because that information is carried through DNA. But how it is processed, it is the gene expression. Okay, it is usually the production of the proteins, and in this example, it's just color pigments. The functional products of most known genes are proteins, or more accurately, polypeptides. Proteins has the other name is polypeptide. Polypeptide is just another word for a chain of amino acids. Amino acids are the building blocks of proteins, just like the building block of matter is atoms and like molecules. Just like this, amino acids are the building blocks. But these amino acids are get together in a formal in a certain order that makes up that special protein. There are many millions of proteins, thousands of different kinds of proteins, but there is only 20 or 22 or 23 kinds of amino acids. But the order determines the type of protein. Just like we have letters in our alphabets. It's a limited amount, like 26 in the English alphabet, for example. But by using only 26 symbols, you can produce millions, thousands, hundreds of words, right? Just like this, amino acids can produce many, many different kinds of proteins. They are just the alphabet of the proteins. Although many proteins consist of a single polypeptide, some are made up of multiple polypeptides. Genes that specifically specify the polypeptides are called protein coding genes. Not all genes specify polypeptides. Instead, some provide instructions to build functional RNA molecules, such as the transfer RNAs and ribosomal RNAs that play roles in translation. So, many genes provide instructions for building polypeptides. How exactly does DNA, direct the construction of a polypeptide? So this is the picture about that. This process involves two major steps, as you can read out in here: transcription and translation. In transcription, the DNA sequence of a gene, okay, is copied to make an RNA molecule. Okay, that's straightforward. You know this. This step is called transcription. Okay, whatever the information in the DNA, encoded in DNA, it is just copied to another one-strand molecule, a ribonucleic acid. DNA is deoxyribonucleic acid. So RNA is just like a DNA, but it's just a single strand. So the information copied to the RNA. Okay, and this step is called transcription because it involves rewriting or transcribing the DNA sequence in a similar RNA alphabet. In eukaryotes, the RNA molecule must undergo processing to become a mature messenger RNA or mRNA. In translation, the sequence of the mRNA is decoded to specify the amino acid sequence of a polypeptide. These individual circles, pieces over here, you see, they're all individual amino acids, but they come together, they make the polypeptide. Okay. The name translation reflects that the nucleotide sequence of the mRNA sequence must be translated into the completely different language of amino acids. So whatever the information over here, it is transferred, or it is just translated into a new language. Okay, during the process of transcription and translation. All right. Thus, during expression of a protein coding gene, information flows from DNA to RNA and to protein. This directional flow of information is known as the central dogma of molecular biology. So this is what called the central dogma. Non-protein coding genes that specify functional RNAs are still transcribed to produce an RNA, but this RNA is not translated into a polypeptide. For either type of gene, the process of going from DNA to a functional product is known as gene expression. Okay. In transcription, one strand of the DNA that makes up a gene, called the non-coding strand, acts as a template for the synthesis of a matching or complementary RNA strand by an enzyme called RNA polymerase. Okay, this RNA strand is the primary transcript. The primary transcript carries the same sequence information as the non-transcribed strand of DNA, sometimes called the coding strand. However, the primary transcript and the coding strand of DNA are not identical thanks to some biochemical differences between DNA and RNA. One important difference is that RNA molecules do not include the base T. Okay, instead of T, thymine, they use uracil, U. Okay. So they have the similar base, which is called uracil instead of thymine, like thymine and uracil pairs with adenine. Okay, all right. After transcription and in eukaryotes, after processing, an mRNA molecule is ready to direct protein synthesis. The process of using information in an mRNA to build a polypeptide is called translation. During translation, the nucleotide sequence of an mRNA is translated into the amino acid sequence of polypeptide. Specifically, the nucleotides of the mRNA are read in triplets. Okay, you see triplets, groups of three base. They're called codons. Okay, each groups of three bases are called codons. There are 61 codons that specify amino acids. One codon is a start codon that indicates where to start translation. The start codon specifies the amino acid methionine. So most polypeptides begin with this amino acid. Three other stop codons signal the end of polypeptide. Okay, these relationships between codons and amino acids are called the genetic code. All right. All right. So translation takes place inside of structures known as ribosomes. The ribosomes are very important, organelles in cells like eukaryotes, and this translation takes place inside the ribosomes. Okay, so ribosomes are, you will see, we will have a simulation concerning production or employing the ribosomes. You will see that in for next coming forthcoming homework. Ribosomes are molecular machines. They're molecular machines. Their job is to build polypeptides. Once a ribosome latches onto an mRNA, okay, and finds the start codon, it will travel rapidly down the mRNA. It's like pulling it and processing, reading one codon at a time, and as it goes, it will gradually build a chain of amino acids in this direction. That exactly mirrors the sequence of codons in the mRNA. How does the ribosome know which amino acid to add for each codon? As it turns out, this matching is not done by ribosome itself. Instead, it depends on a group of specialized RNA molecules called transfer RNAs or tRNAs. Each tRNA has a three nucleotide sticking out one end, which can recognize base pair with just one or few particular codons. At the other end, the tRNA carries an amino acid, specifically the amino acid that matches those codons. There are many tRNAs floating around in a cell, but only a tRNA that matches base pairs with the codon that's currently being read and can bind and deliver its amino acid cargo. Once a tRNA is snugly bound to its matching codon in the ribosome, its amino acid will be added to the end of the polypeptide chain. Okay, once the matching is done, that is the process. The process repeats many times, with the ribosome moving down the mRNA one codon at a time, but this happens very quickly. A chain of amino acids is built up one by one. Okay, this is attached to this one with an amino acid sequence that matches the sequence of codons found in mRNA. Translation ends when the ribosome reaches a stop codon. There is a codon which tells that you should stop and releases the polypeptide. Once the polypeptide is finished, it may be processed or modified, combined with other polypeptides, or be shipped to a specific destination inside or outside the cell. Ultimately, it will perform a specific job needed by the cell or organism, perhaps a signaling molecule, a structural element, or an enzyme. Alright, so let's make a summary. So DNA is divided up into functional units, they are called genes, and which may specify polypeptides, proteins and protein subunits, or functional RNAs such as tRNAs or rRNAs. Information from a gene is used to build a functional product such as protein in a process called the gene expression. So the gene expression is that the process that the information turns out to be a specific product. A gene that encodes a polypeptide is expressed in two steps. In this process, information flows from DNA to RNA and to protein, a directional relationship known as the central dogma of molecular biology. One strand of the gene's DNA is copied into RNA. In the eukaryotes, the RNA transcript must undergo additional processing steps in order to become a mature messenger mRNA. So this process is called transcription, and the nucleotide sequence of the mRNA is decoded into specify the amino acid sequence of a polypeptide. This process occurs inside a ribosome, an organelle inside the cell, and it requires adapter molecules called tRNAs, and this process is called translation. During translation, the nucleotides of the mRNA are read in groups of 3. They're called codons. Each codon specifies a particular amino acid or a stop codon, a stop signal. This set of relationships is known as the genetic code.